DNA is an amazing but really simple molecule. And so there is this question, how can DNA code for so many different proteins if it is such a simple molecule? And really, when we get down to it, DNA is made up of 4 different things, 4 different nucleotides that contain 4 different nitrogenous bases. The way that this is accomplished with DNA is that it's going to operate very similar to the alphabet that we have for our language. So if you consider our alphabet runs A, B, C, etc., all the way to Z. And so that right there is just 26 different letters, but those 26 letters can be arranged in a number of ways, a number of combinations, and because we can arrange them in so many combinations and in so many lengths, we can come up with really an unlimited number of words from those same 26 letters. DNA is going to operate very similarly because DNA, it has 4 different nucleotide bases. So within DNA, we do have the 4 bases A, T, C, and G. And these are like the letters of DNA. So DNA has 4 letters, and those 4 letters can be arranged in different ways. So we will arrange them to make words, or what will be the equivalent of words. And really, when we get down to it, what we need our DNA to say is we need it to be able to specify the instructions to make the proteins that operate within our cells. Our DNA is going to be used to make proteins, and our proteins are made of 20 amino acids. And those 20 amino acids will be joined together. We'll have ribosomes that are responsible for joining those together, and the proteins will be made of various lengths. So we might have a couple hundred to maybe even a couple thousand amino acids joined together. But it is the instructions in the DNA, the actual order of those nucleotides, that is going to specify exactly which amino acids to connect together. So our nucleotides, we said we have 4 of them, and they will be used to make words. Well, the words are going to be called codons, and each codon will tell the cell a specific amino acid. And AA stands for amino acid right here. So when we read those nucleotides, if we were to read them just one at a time, so we had, say, only one that was specifying the instructions. Well, if we only read one at a time, that really only gives us 4 combinations or 4 words, which is not enough because, as we mentioned, we need to code for 20. So 1 letter at a time is not going to work if our words were made up of 2 letters at a time. If we had that and we consider that each is 2 letters long, and in each one of those blanks we could have any one of the 4 letters, A, T, C, or G, that would give us 16 possible combinations or 16 words, which again is not enough because we need to code for 20. So instead, the way that this is going to work inside of our cells is we will read them 3 at a time. So we will have 3 blanks, and those 3 blanks will give us 64 combinations. And so again, in each one of the positions we can have A, T, C, or G. And when you look at all the possible ways that we can arrange that A, T, C, and G together, we do get 64 different words. We're going to call those 64 codons. So really reading them 3 letters at a time, that's the minimum codon size that would work in this case for the 20 amino acids that we do find inside of our cells. The picture that we're seeing right here is what we call the genetic code, and the genetic code is really just showing us all the various 3-letter combinations that we do have, and really what the meaning of those is inside of our cells. Now notice in this genetic code table that we don't see T's represented, we see U's represented, and that's because the codons are found in RNA, but the RNA that we have is really made from DNA instructions. So the codons on here, they do show us there's 64 of them, most of them, so every one of them except for 3 is going to code for an amino acid, and there's certainly some redundancy there. And what we mean by redundancy is that we can have multiple ones, and here we actually see 6 different ones that can code for this same amino acid, which happens to be leucine in this case. So sometimes there are multiple codons that code for the same thing or have the same meaning, and then in other cases there might be just one of them that codes for a particular amino acid, and an example of that would be located right here. Okay, so that's our genetic code table. All of the organisms that have been discovered to this point contain DNA that's made up of 4 different nucleotides. And those 4 different nucleotides contain the A, T, C, and G bases, and those are rearranged in combinations of 3, so codons that have 3 letters composing them, and that's used to specify the 20 different amino acids that make up our proteins. Now suppose we had a different situation. Suppose that scientists discovered a new species, and that species contains a new form of DNA. And in this particular DNA, it only contains 2 different nucleotides that have different nitrogenous bases. And the proteins in this species are composed of 40 different amino acids. So the question is, what would be the minimum codon size that would be needed to code for the amino acids found in this particular organism's proteins? So if we look at this, now we're seeing that the only letters we have maybe are A and B. So we have A and B, and then these are going to be read in some type of combination, and we need to get 40 different amino acids specified. So the 40 different amino acids, this would be like our words that we need to create. We need to be able to create 40 different words. And the nucleotides, that is the letters of our alphabet that we will be using. So we want to know, how big do the word sizes have to be, or how big do the codon sizes have to be? Well, if we read them typically in 3-letter combinations, which is what is our normal codon size, well, remember that we only have A and B here. So we could have either A and B in the first spot, we could have either A or B in the second spot, and we could have either of A and B in the third spot. And if we had that and we multiplied those together, we would only get 8 combinations. So 8 combinations is not enough to specify the 40 different amino acids that we actually do have in this particular organism. So instead of reading them 3 at a time, we have to read them in bigger sizes. So if we read them 4 at a time, we would have another blank right here, and that would give us 16 possibilities. Again, that's not enough. If we had another blank there and we multiply that all together, we would get 32, which again is not enough because we need to be able to code for at least 40. So if we put another blank here and you went through and you multiplied all of this, we would get a total of 64 combinations here. So 64 words or 64 different codons that we could code for the 40 different amino acids with, and there would certainly be some redundancy there. But what this means is that our minimum codon size is really going to be 6, because that's the smallest number that we can read together to give us at least 40 different combinations. Now, if the codons were bigger than that, if they were 7 letters or 8 letters, that would certainly cover the 40. But what we're looking for here is the minimum codon size that could be used to code for the 40 different amino acids in this particular species.